Viventia Bio Inc. is a biopharmaceutical company specializing in the discovery and development of a new generation of targeted therapies in oncology. Viventia Bio's targeted protein therapeutics, or TPTs, utilize cancer-specific protein molecules to seek out and deliver a highly potent protein payload to tumor cells. To ensure better safety and clinical benefit over current therapies, Viventia Bio has taken a rational design approach to engineering their TPTs by incorporating distinct drug design features. 2 TPTs have successfully completed Phase 2 trials in areas of high unmet medical need: Vicinium for the treatment of bladder cancer, and Proxinium for the treatment of head and neck cancer. Using our immune-driven antibody platform, Viventia is creating a portfolio of new TPTs directed at proprietary oncology drug targets. Debuganin, Viventia's proprietary de-immunized payload, will allow repeated systemic use of these new TPTs. Viventia Bio, targeting cancer with power and precision.
Hello, and welcome to Viventia Bio. I'm Glenn McDonald, Chief Scientific Officer of the company. Today I'm going to take you on a tour of the research and manufacturing facility to introduce you to the drug development process from beginning to end. This file represents the end product of our drug development process. To understand how we get to this end, let's start at the beginning in research and development. At our research facility, we take antibody fragments and we arm them with highly potent cytotoxic payloads. These antibody fragments target the payload to the cancer cell, resulting in the death of the tumor. To accomplish this, we use recombinant engineering technology. Clinical candidates are evaluated using state-of-the-art techniques such as flow cytometry. Those molecules deemed clinically relevant are then transferred to manufacturing.
This is where manufacturing begins. Hi, I'm Arjun Premsuk, Director of Process Development and Manufacturing. I'd like to give you a short tour of our fermentation development lab where we achieve high cell density cultivation of E. coli cells. In this lab, we have several different bioreactors that we use for fermentation development. We have a 2-liter bioreactor, which we use initially for fermentation and to identify basic fermentation conditions. We would then scale that fermentation process to our 15-liter scale for further optimization, and finally to our 150-liter scale prior to transferring that process to our cGMP fermentation suite for clinical and commercial manufacturing. This is our cGMP fermentation suite. It's a controlled environment, hence I'm gowned up in clean room coveralls, which allows me to work in this area. Firstly, I'd like to show you our primary seed culture room, where we would retrieve a vial of cells, either from the master cell bank or a working cell bank, and use this for inoculating a 2-liter shake flask. This is known as our primary seed culture. This culture is then grown in an incubator shaker to predefined specifications prior to using that culture to seed our 35-liter bioreactor. Once predefined optical densities are achieved, we would then use the culture from this reactor to seed either our 500-liter bioreactor or our 1500-liter bioreactor. Fermentation is then performed for around 4 to 5 days, and the product at that time is secreted into the culture media. To separate the cells from the culture media, we then use a dish stack centrifuge, and the cells are discarded and the product in the supernatant is collected. We then process the supernatant using microfiltration and then concentrate the microfiltered supernatant by ultrafiltration. This is an example of our microfiltration system, and then we finally transfer the product over to our ultrafiltration system where diafiltration concentration is performed. in order for it to be purified in our pilot plant purification area. We are going to enter our cGMP purification suite. In this suite, we have several areas that we covered, including components preparation, where we do all the washing, sterilization of the components, the bottles, the caps, and so on that's used in the purification and the fill finish. We also have a buffer preparation room, a sterile fill finish room, a purification room. This is our wash-up area where bottles, caps, containers, closures is prepared for purification. Then we have our buffer preparation area where all the weighing of salts used to prepare the buffers is done. We have a Wi-Fi outlet for water, we have a flow balance, and we have an overhead mixer for mixing large volumes of buffer that we use in the purification process. In the purification room, it's a class 10,000 area where the product from the fermentation suite is transferred in bags into this room. Using the bioprocess skid, we then use multiple columns to purify the product to meet certain specification. These chromatography steps include affinity chromatography, ion exchange, hydrophobic, and so on. Once the product has been purified to meet specification, we would then formulate the product using a tangential flow system. This allows us to put the product at a certain concentration and formulation to allow for long-term storage. The product is then transferred into our sterile fill finish area where it's vialed, labeled, capped, and quarantined by QC until QC release testing is completed. This is our quality control lab where final release testing of the product is done. Once the product meets specification, it's released into the control of quality assurance and placed into long-term storage conditions. That's the end of the process, and that's the end of the tour.
Integrated recombinant drug development from beginning to end. Research and manufacturing all under one roof. That's Viventia Bio.